Well, thanks everybody for coming. I wanted to talk a little bit about some work that we did for the last 2 years. I've done it before as well, and I'll give you some background on it. But she couldn't be with us today, but this is our newly minted master's student, Chantel, who undertook most of this research. Anyway, she's now back in San Diego, so she couldn't come. I think we all kind of appreciate that we're sort of in this field of looking at residential tick control, and that the people at the home side of it all, their mailbox is flooded with things like this. They don't know what they're spraying to treat for ticks. And even after you ask the companies that send out those colorful flyers, you don't exactly know what they're treating for ticks. But generally speaking, the big selling point is natural products, minimal risk, not needing to be approved by EPA. And because of that, we don't really know how well they work. And so I realized this years ago, and with support from CDC, we created a project that would sort of in a very robust way try and assess and screen natural products for their efficacy against nymphal stage black-legged ticks. And this is just what that looks like. And our field design, you can see we're interested in knockdown and residual efficacy. So knockdown is sort of spraying on the ticks that are in there, and then the residual is done 2 weeks later. So a 2-week residual. We've sprayed the plot, but then we add ticks to that just to see if it had any residual activity. And so all of these are individual treatments. We replicate everything 5 times. We're able to use 60 ticks. So it's pretty robust. Like all of you sampling in people's yards, you realize that if you'd have to do a lot of work in order to get a statistically sound assessment of treatments. Maybe the main team has done a better job with that. And I know Nita has some success too, but it's a lot of work. This is a lot simpler. So we've looked at it this way is sort of the sampling plan. So we deployed the ticks the day before we get started. We apply the products on day 0. Then we sample on these 3 days for the knockdown. And then we deploy ticks again at the beginning of the residual period. And then we look at a 2-week residual efficacy as well. This is how we do the sampling with a sort of a customized little flag. And the way it works is you sort of pump it up and down for 10 seconds, and then you look, and then you take the ticks off, and then you do it again until you don't get ticks for 3 sample times in a row. And then you can move on to the next thing. It's a little bit tedious, but it's not that bad. You get to sit on a little stool in the woods. It's not too bad. To get to this point, though, we have to raise ticks. And so for our project, we probably raise 10,000 nymphal ticks every year for deployment in the woods. And it's a big job to raise that many, but also the day that you're getting ready to put them out, you have to count them all out as well. So sort of 4,000 or 5,000 ticks to deploy. Takes a lot of student help. And then we've been blessed to have a lot of good students that have helped us with this in the summer. So this is putting ticks out. You just sort of knock them into the arenas tick habitat right on similar to what's on the edge of people's yards. And then we cover them. It's not to keep the ticks in, but it's to keep animals from taking our ticks out accidentally. Then the day of the spray, we mix the sprays up in these little vials, and we have a device that's powered by CO2, and it puts out a fairly typical spray in this small area. We have this spray shield that just sort of keeps it all there because you could see that everything's sort of close together, and we don't want any cross-contamination because we sort of mix within the array. We mix the different treatments, and so we have all these flags. It gets a little complicated for a guy like me to try and figure out what I'm sampling, but that keeps us kind of dumb to the whole thing. You don't know exactly what's in there until you sort of break the data down later. This is what the sampling looks like. And this is a team that was doing a study. And so the interesting part is when you sort of accumulate all of the data and sort of analyze it. And this is the first year of the first contract that we had with the CDC, and the green products are all natural products. And then water is one of the controls, and Talstar bifenthrin is the positive control. And what I hope that you don't have to look at all of them necessarily. I hope that you'll appreciate that some things work really well and most things don't work really well. If you remember the CDC study that was done that showed, I think Nita was part of that as well, that if you had achieved 60% tick reduction, you still didn't really impact people's tick encounter rate or the level of disease prevention. And so 60% seems to be not good enough for residential tick control to be very valuable. But you can see that bifenthrin, in our case, always gave pretty good control. And some of this natural pyrethrum actually had pretty good knockdown control and a little bit less residual. But one product, I'll get to this in a minute, NEWVEC, we've all heard of it. Most of us can't say it right. This was an early formulation that the CDC had that was derived from Alaskan yellow cedar. It's also now synthesized as a food additive and being potentially prepared for marketing as a tick control. And you could see it gave pretty good knockdown control in this particular early phase. This was back in 2012 to 2015 that we did this work. Then we had another contract in 2020 during the pandemic from the CDC, but we were able, since it's outside work, we were able to do that. And I learned not to put the product name because some people take offense when their product doesn't work. And so I just put the active ingredients here. And you can see we had granular products, we had liquid products. But again, I rank them toward sort of in terms of efficacy. And you can see most of them are not really ready for prime time when it comes to tick control. Here are the controls down here, the positive controls, all with high kill rates, pretty good residual rates. But the best one in that group had pyrethrin in it as well, which is natural, but still not that great. So I think that a lot of these are all commercially available products, and this is what people are getting and putting on their yards, and this is likely the level of control they're getting. So it's a little bit problematic, I think. Well, so I wanted to talk a little bit about NEWVEC. So CDC had a particular effort to move this along, and there was a Swiss company called Evolva, and a few years ago, the EPA did approve NEWVEC as a pesticide, in particular as a mosquito control repellent, and also there was some interest in developing it as ticks. But I think our work is probably one of the first ones that is using the Evolva product to test its efficacy against ticks. And so here you can see sort of an array of the information that we got from that. So we got 3 products from them. We called it Group A, Group B, and Group C. And we got it in 2 concentrations, 10% and 3%. And Group A, I think, was designed to be might have been a surface spray. Group B was supposed to be a repellent. And Group C was sort of mixed as a yard spray, like a hose end yard spray kind of a thing. And so you can see that it's dose dependent. The 10% generally was much better than the 3% ones. And group C and group A both achieved knockdown kill rates pretty comparable to the tau star right there. One thing you might notice, this one called placebo, we weren't really told what was in it, but we knew that it didn't have nucleotone, but it looks like whatever they put in there also had some killing as well. One of the exciting things that I think we did, so we also have just in the last few years started getting Lone Star ticks in Rhode Island, and so we wanted to see if these products might be effective against Lone Star ticks as well. But any of you that know about Lone Star ticks, they're far more aggressive than black-legged ticks, and so we figured they were going to want to crawl out of our little arenas. The black-legged ticks, pretty much most of them end up just staying in the little arena. But so we needed to devise a system where we could potentially contain the Lone Star tick nymphs inside. So we sort of made a double-tiered arena, and we used some sticky stuff that would try and help keep them in. We didn't really know if it would work or not, but that's what a good graduate student's supposed to be there doing, trying to figure stuff like that out, right? And so here's sort of the base arena down here, and then sticky stuff started up here. Of course, we had to take them off when we did our sampling because it would make the flag all sticky and everything. It's a little bit of pain, but you can see some of the nymphs, and it kept the nymphs in well. We found probably less than a handful in the sticky stuff that they would go up to the sticky stuff and then turn around and come back down, I guess. So it actually worked better than we expected. We had raised about 1,000 Lone Star tick nymphs for this work, and that allowed us to have enough to have a positive and negative controls, and also to test both products from Group A and Group C. And we just did the 10% there to see if it had any efficacy at all. And you can see the results are shown here. And it seemed like it had reasonable knockdown efficacy even against Lone Star ticks. So that's good. The Group C, which was the one designed for the yard treatment, gave about a little over 90% control. The last thing I wanted to talk a little bit about, we had a group at URI, Steve Alm and Matt Kieswetter. He's a chemist, and they were interested in a problem of varroa mites affecting honeybees. And so one of the big issues with backyard and people wanting natural products is they think they don't want to kill honeybees. And so it'd be nice to have a more bee-friendly tick killer. And so Matt, as a chemist, had this brilliant idea. This is a varroa mite on a honeybee. They had trouble sourcing, the reason I got involved, they had trouble sourcing varroa mites, and they thought, oh, nymphal stage ticks are about the same size, and I have lots of those. Could we just use that as a surrogate? And so I said, that seems great. They hadn't thought of this whole bee-friendly tick killer thing, but I thought, well, while we're giving them ticks, we might as well figure out if we can see if there is an effort there. And so what Matt appreciated is when people develop insecticides, when these companies, they actually are looking for broad spectrum control, right? That's where the market's going to be. Let's get something that kills, and they do these various screens with all kinds of organisms, and they actually toss out the ones that have differential control. But we're more interested in differential control now. And so he had a student who synthesized some derivatives of fluvalinate. Fluvalinate is a synthetic pyrethroid. And so she came up with several. She made 5 initially, and we screened them. So this fluvalinate, TF there, is the fluvalinate structure. And then these others, they're called KP because the student's name was Cassie, and her last name started with a P. And so we did trials in the laboratory to figure out a lethal concentration 50 dose. And we did that by looking at, screening a whole bunch of concentrations with 10 ticks per vial and replicated 4 times, and we ended up with 2 products, KP41 and 75, that seemed to have a fairly good differential kill, less toxic to bees and still killed ticks. What I can show you here are the results from the lethal concentration 50 work. So you can see here's tolfluvalinate. Here it is against bees. You can see there's a pretty good difference between the kill rates on the concentrations. And this was, both KP75 and 41 appeared to have reasonable differences, but the KP41 actually, was 20 times less toxic to honeybees than fluvalinate, which is, I'd say that's bee friendlier at least. And so it was still toxic to ticks. And so we needed to see what happened if we put them out in nature. And so we did that in the second year of Chantel's study. We tested the KP products at both 0.1 and 1 milligrams per mil, just to see. And you could see, again, there's a dose response. The 0.1 seemed to be much better, but all of the KP products seem to have reasonable tick control capacity, at least, when compared to both this Maverick, which is the fluvalinate product that's available for spraying backyards by PCOs, and Talstar. So fairly comparable results. In moving forward, I think we have been able to say that the 10% Nucotone seems like the way to go. Company and discussions with them would prefer thinking about the 3% one because it's more cost effective to sell to people. I guess it's expensive at this point, but it was certainly a little bit less effective than the 10%. And the LD50 for the KP75 is not all that different from the LC50 against bees, but the 41 is got a almost 20%, 20 times difference in terms of its efficacy against bees, but the efficacy is a little bit low for the lower dose one. So it was only around 60%. So we may have to make suggestions going forward that we could maybe either look for other products, and I know they're developing some additional things to test, but also just work with the concentrations to see if we could boost it up to get a little bit better tick killing without harming the bees. So that's where Chantel left us with this work, but I think it was pretty informative and hopeful. I'm a little bit hopeful that we can move forward with some actual natural products that are good tick killers.